Welcome to our annual Fall College Science Scholars Research Symposium. We have 7 speakers who will be giving summaries of their research that they've been spending quite a bit of effort on for the last many months, sometimes several years. And I think you'll find these very impressive. We're very excited to be here and have our fine young scholars give presentations. I want to introduce a couple of people before we get started. First, my 2 co-directors, one who has a camera to his face, Jim Demas. Yes. And the other who is munching on a sandwich. They're busy. Howard Epstein. And without these 2 gentlemen, this program wouldn't run as smoothly as it does. So I also want to recognize some special guests we have in the room. Mary Beth and Jeffrey Lowe, who have provided endowed support to our program, and we are very grateful for you and your presence today as well. Thank you. Our first presenter is Artie Purohit, who is a second-year CSS student, and she is a Lowe College Science Scholar, and she's going to tell you more about her work. Her mentor is Dr. Ali Gumar in the Department of Biology in the topic area of neuroscience. Take it away.
Thank you. So I'm Artie. Thank you for coming to this symposium. So I'll be just presenting my research and data that I collected this past summer, where I was here for 10 weeks. And like Karen introduced, I do work in the UVA lab, which is in the Department of Biology. Our lab is focused on the role, its overall arcing project is the role of dopamine in circadian entrainment to reward. It's a mouthful, and I'll explain more of it as we go along. But essentially our lab is a neural connection lab. We try to figure out what areas in the brain are being connected from where and what areas project to what positions in the brain. So I just wanted to start out with introducing what dopamine is. It is a neurotransmitter, which is a chemical signal that is used to transmit signals from one neuron to another. So this is the target neuron. This neuron is a dopaminergic neuron, so it's producing dopamine. It's released across the space called the synapse and picked up by these receptors on this target neuron, which then induces a neural impulse. So this is how neural impulses are sent from one neuron to another. There are several dopamine systems in the brain, but the one that I will be focusing on is the one that is involved in reward-motivated behavior. And then the other point of focus is the suprachiasmatic nucleus. This is a region in the brain which is located right here on the bottom. So this is a mouse brain. Here's the front of his brain. Here's the back. So if you were to cut right down the middle and we're looking at it, this is what you would see, and it's very, very to the bottom. So the suprachiasmatic nucleus, or what I would refer to as the SCN, is our master biological clock. So it is what keeps all our biological systems in secret. The rhythms are endogenous, which means that if I were to stay in complete darkness, I would still be able to regulate these circadian rhythms. But they're regulated, like I said, by environmental cues, and they can be photic or not photic. And this is what's referred to as entrainment. So the regulation is referred to as entrainment. And the example that I like to provide for photic input regulation is jet lag. So if I were to travel from here to, let's say, across the world in Russia, it would take time for my body to adjust to the new light-dark cycles over there, but I could do it. It would take time, but I would be able to do it. But what my lab focuses on is this non-photic input. And so the example I like to provide for this is research has shown that mice, which are nocturnal animals, so they will be sleeping during our day and then awake during our night. But if you give them food only during our day, they can actually shift their circadian rhythm to wake up when the food is provided. So what is causing that? What is regulating that? So the central premise that we focus on is that the fact that the SCN has receptors for dopamine, the D1R receptors, so these are what bind to the dopamine molecules. And then the second, that the SCN neurons should have light-dark rhythmicity, so they should be more or less active during the day and vice versa at night. And I want to just see if I can play this video that really visualizes what's happening. So you can see that the activity of the neuron, so this is playing really slow, but the activity of the neuron increases and diminishes, and then this is over time. There. So going on to my own personal research. So I use 2 main techniques in my research, which are very novel techniques, so I thought it would be a good idea to just explain them. So one of them is what are called DREADs, or designer receptors exclusively activated by designer drugs. So these are receptors that have lost their affinity for natural ligands. So they will not bind to anything that is naturally present in the body. They can only bind to these designer drugs, such as clozapine oxide, which I will refer to as CNO. And essentially what happened was Q recombinase, which is a biological cell biology technique, was used to target these receptors only to the SCN neurons that had the D1R receptors. So essentially, if successful, I could inject a mouse with CNO, and then the activity of the neurons that had these drug receptors would either increase or decrease in response to the CNO. And then the second technique that I used were these recording implants, so which were surgically implanted on mice, as you can see here, and they're essentially an electrical board that electrodes were threaded through, and they would poke out here, and then they would be surgically implanted all the way down into the SCN. So I could record from individual cells at the SCN neurons. So the major benefits for this technique is that the mice are freely behaving. Once you hook them up to a data acquisition software, they can walk around in their cage and I can still record all the neural impulses. I can even turn the sound on, I can hear the neural impulses firing. And then if the surgery went very well, I can record from the same mouse for 1, 2, 3, 4 months, however long I want to do it. And then the second main point is that there are screws here, so I can screw down the electrodes so I can reach deeper and deeper into the brain and record from more cells. So my experimental design, I've gotten some bits and snaps of it, but essentially what it was is I had implanted 4 mice with electrodes that I was saying I targeted at the SCN. They were then entrained to a 12-hour light, 12-hour dark cycle, which means so that from 12 a.m. to 12 p.m. the lights were on, from 12 p.m. to 12 a.m. the lights were off. So they were entrained to this cycle for a couple of weeks just so that their activity was at a baseline level. And then I would activate the dopamine system and then monitor the SCN activity. So I would record from the SCN in the morning and in the night. So I should see a difference if I'm recording from a rhythmic SCN neuron as opposed to the morning and the night. And I'll show you some data that supports this. And then I would also record in the morning and then inject CNO, and 30 minutes later I would record again to see if there was a CNO response of the neuron. So the results were there was 4 mice injected, 1 head stage had wiring issues, so unfortunately I could only gather data from 3 of them, but that was not an issue for this particular research because it was just a proof of principle. I just wanted to see if this protocol would work. So several units or cells had data at rhythmicity, and then I'll explain that on the next slides. So this is just a point of reference to see how well my targeting was. So the SCN is located here, and then these are my 3 mice, and you can see that this is a tetrode extending, and this was a perfect targeting of the SCN. This was our accidental negative control because the virus or the drug receptors didn't get down there, and then this one is focused, is zoomed up picture of this region. So it went pretty well. So this is what data analysis looks like when it's inputted into MATLAB, which is just a computational software. But the 2 main things I want to point out are these firing rates, which is how often the neurons fire per second, and then the shape of the action potential. So this is an example of one cell that I recorded from. You can see that there was an increase in the baseline day and night activity. So it went from 5.94 hertz, or firing per second, to 10.9, or 10.4. And then I inject CNO, and immediately, 30 minutes later, the activity is reduced by half. And you can also see that in this cell. There were a lot of cells, but these are just 2 examples that are provided here. It went from 0. So this is baseline day, baseline night, and you can see that there was a significant increase. You inject CNO, and it's reduced by half. So what is the significance of this? So this is essentially a proof of principle study. It shows that this system does work. And the future steps that I can take is to repeat the experiment with more mice, proper targeting, but then also I can induce dopamine production in different regions of interest in the brain and see if the SCN neuron activity is increasing or decreasing. And if it does, then I know that it's being innervated by those regions. So at that, I will leave it at that, and so hopefully my future study will be looking at that in the future. Thank you. Many thanks goes to the generous donors of the College Science Scholars Program, which makes all these awards possible. And special thanks to Mr. and Mrs. Lowe, Mary Lowe and Jeffrey Lowe, and of course, Ryan Grippa, who's a graduate student in my lab, and Andre Rainwater, who's our lab tech. I'll leave it at that. Any questions?
Yeah, we have time for questions. So it seems like it would take a long time to surgically implant all of those electrodes into the mice as humans.
It actually takes a longer time to actually build the electrodes because you actually have to, I myself have to use a microscope and use threading and thread the electrodes in. The surgery takes about an hour if you're experienced, and I usually just let my grad student do it because he'd been doing these surgeries for a long time. So it takes about an hour to do the surgeries. What happens to the mice when you're done with the experiment? Do you remove the device? Yes, so you can, so they're cemented with dental cement, and you can just pop them off, and the animals are perfused or sacrificed, and you saw the brain slices, so their brains are sectioned to see if there was proper training of both electrode and the drug receptors.
Other questions? All right. Great. Thank you.
No. Science is always in process. we have one tweaking that's taking place right now.
Distance to volume.
It's so exciting. Okay, next we have Sarah Wyckoff, who is a second-year college science scholar, and she works with Dr. Stephen Solden in laboratory medicine at NIH in clinical mass spectrometry. She'll tell you more about her topic.
So as Karen said, my name is Sarah Wyckoff. I'm second year in the College of Science Scholars. This summer I interned at the National Institute of Health's Department of Laboratory Medicine in their mass spec unit, and what I studied was free vitamin D3 analysis, so I state-of-the-art in liquid chromatography, tandem mass spectrometry technology. Okay. So the future belongs to the free. Free hormones are not bound to proteins in the blood and are therefore the biologically active agents that are able to bind to target sites on receptor cells in order to initiate a response. It's thought that by looking at the different concentrations of proteins in the blood and the free concentration, it varies from individuals to individuals and with different clinical conditions. And it's thought by measuring the free that you'll get both a better diagnosis and sort of idea of how to treat people. A particular interest is 25-hydroxyvitamin D3 because of vitamin D sort of implications with the immune system and with bone health. So far, however, only reference intervals exist for the total measurement, which includes the bound proteins and the free, and those a reference range is about 30 to 100 sort of nanograms per milliliter. That's considered optimal for health. However, again, this is very sort of general and isn't widely accepted by all. So far, it's thought that maybe a reference interval for the 25-hydroxyvitamin D, the free version, would maybe give a sort of tighter reference range and be better indicative of optimal vitamin D levels for health in the human body. But up until this point, no method has been able to both accurately and consistently measure free vitamin D. And this has sort of been a problem because we haven't been able to look at it. All the methods that exist today have really been sort of calculation-based and haven't been looking at the analyte itself. So in this study, what we did is we developed a way to quantitatively measure and analyze free vitamin D through liquid chromatography, mass spectrometry analysis. And with this, we looked at 13 patient samples and developed a tentative reference range from about 17 to 100 picograms per milliliter, which is much tighter than what exists already. And it was thought that by this would open the door to a lot of different clinical applications and an optimization of treatment in various cancers, bone diseases, etc. So what our hypothesis was at the beginning was that free vitamin D is indicative of the effect of vitamin D on the human body. And that by free vitamin D, it plays a vital role in human health and disease, and that its concentration and quantification of that will enable connections to be made between that and certain diseases and conditions. In addition, the measurement of total vitamin D, which is right now the current standard, won't necessarily correspond to free vitamin D measurements, and it won't necessarily be an indicator of health. So our goals for the experiment are to employ state-of-the-art liquid chromatography, tandem mass spec technology in the analysis of free 25-hydroxy vitamin D, and then to develop a tentative reference range for what's normal for patients in the NIH population. And then in our final goal was to compare total 25-hydroxy vitamin D measurements to both the free and percent free to look for any correlations that might exist. So here are results. Unfortunately, I can't talk too much about the method that we use. While that was a large part of my research, it's actually sort of in the process of being approved for a patent. So that'll be coming out in the next couple months or so. However, what we did get is we, like I said, use mass spec. And essentially what that allows you to do is separate compounds based on their polarity through a liquid chromatography, and then they're injected into a mass spec machine, which sort of detects different particles based on their mass to charge ratio. And the strength of the signal is indicative of the amount of sample present. So by integrating the peak, as we did, sorry about that, at the top one, you can find the sort of relative amount or quantitative amount that's present in the sample. So what's really important about this top peak is the fact that it's there. In a lot of other studies that were done, it wasn't showing up, and that was the problem. It was lost somewhere along the sort of development and preparation process. So the fact that we have a nice sharp peak at the correct retention time with very little background noise means that we did it right and that it's sort of easily quantifiable. The bottom shows the internal standard, which is of the same structure as free vitamin D, however, different molecular weight. So you're able to identify it separately using the mass spec. And what you do is you use a sample of no concentration, and then when you go through, you compare the concentration that the mass spec calculates to the known. You're able to develop a calibration curve to check your sort of unknown quantities against that and account for any sort of error or issue with the device. So it really gives you an accurate and consistent measurement no matter what. Here is an example, here are the 13 patient samples that we took, 1 through 13. And what's important is we look at the ranges here. So for the total, it was 19 to 105, but for the free, it was 0.1 through 0.17. As you can see, this is much, much tighter. So like potassium, if someone was outside of this range, it would be much easier to pick up because it would be sort of instantly noticed as abnormal. We look too at the samples 8 and 10 because while they have very, very different totals of 20 and 105, their frees are fairly similar. So while someone with 20 might be considered deficient or low in vitamin D, their free levels are relatively high comparatively. And so they might not be suffering from any of the symptoms of vitamin D deficiency, while someone with 105 may not be as well off as we think. Here's a graph sort of illustrating the total vitamin D levels against the free. As you can see, there is relatively little correlation. However, we might have to add more patient samples, and we're working on doing that in the future, and this is just sort of a preliminary study. And then here is the total free versus the percent free. And I think it's important to note that it actually, from our samples so far, shows a decreasing trend, which is interesting because it means that the more vitamin D that you have in your system, bound or otherwise, might not necessarily mean more biologically active and ready for use. Future directions. So what we hope to do is examine the role of free vitamin D in bone and immune health. Because what this study did at this point was we wanted to see if we could measure it. And we could, which is exciting. Now it's sort of like, what do we want to do with it? And one of the experiments that they're thinking of doing, and that they've already started, and I helped with this summer, was examining the relationship between free 25-hydroxyvitamin D and parathyroid hormone in different patient populations in NIH. And so that's an ongoing process, and they're probably about halfway finished. I checked in over fall break, and it's going well. So it's exciting because vitamin D has been linked to a lot of diseases and conditions lately, such as Alzheimer's, osteoporosis, et cetera, and different cancers. And all of this research is great, but what they've been doing is they've been looking at the total. So it might be interesting to sort of go at it from another perspective and looking at the free and seeing if the relationships still exist and if they're even stronger, and maybe we can sort of treat it that way. Here's an example of the poster that we did this summer. So all of the interns at the National Institute of Health create posters and present at the end of the summer. So this was mine. And I'd like to thank here are references that were used. A lot of the methodology was based on some of the techniques I mentored developed over the years. And so he's been building on it for a couple of years, and it's sort of all culminated in this project, which was an exciting time to be there. And then I would like to thank my mentor, Dr. Stephen J. Solden, Bernie Goulden, and all those involved with the summer internship program at the National Institute of Health. I'd also like to thank the College of Science Scholars Program for supporting our scientific development here at UVA. So, thank you.
Okay, we have some time if anybody has a question.
I think I missed this, but what's the difference between like vitamin D and free vitamin D?
So vitamin D itself, I guess, is sort of a more general term, and I did flip back and forth a couple times. Free vitamin D refers to the hormone that isn't bound to any proteins in the blood. So it's freely circulating and it's able, therefore, to fit into receptors on target cells. Bound vitamin D is bound to vitamin D binding protein. And there are 3 different types that people can make, and it'll vary genetically between person to person. And when they sort of are bound to the vitamin D, that's what the total usually looks at. So the total will measure both the bound and the free levels.
Other questions? All right.
Thank you, Sarah.
Thank you.
So our next speaker is Jessica Dean, who is our 3rd-year College of Science Scholar student, who is also given a Summer Scholar Award by us to do her work this year. Actually, the 2nd time she's been awarded a Summer Scholar Award, so we're very proud of her in that regard. Her mentor is Professor Linda Columbus from biochemistry, and the topic area is structural genomics.
Hello. So, I am a 3rd-year CSS student, and I have been working in the Columbus lab since my summer after my 1st year, and I'm here to talk to you about my project. So I'm working on the co-crystallization of the thermoplasma acidophile nucleoside kinase, hereafter abbreviated as TANK for clear reasons. And basically, protein misannotation is a bigger problem than most people think. Misannotation meaning that ideally, the situation would be where you look at a protein and you predict its function based on its structure alone. However, we're not good enough to do that yet. So there are actually up to 80% of proteins in some public databases are estimated to be misannotated, and that can obviously cause lapses in accuracy, and that can lead to waste in time, man-hours, and money when it comes to research. So why am I studying this somewhat esoteric protein? Well, our overall goal here is to improve our understanding of protein structure, function, and relationships. And under that umbrella, that overarching goal, TANK is a really wonderful case study because it has 2 anomalies that make it very interesting. First of all, it looks like a ribokinase. However, when a student in my lab did some kinetic assays on this protein a few years back, she found that not only did TANK not bind to ribose or phosphorylate it, it actually is a nucleoside kinase, and so it interacts with nucleosides rather than ribose. Secondly, TANK is also promiscuous, or it has broad range specificity, and that means not only does it bind to adenosine, but it also binds to cytidine, to inosine, and to guanosine. And so that's a very rare trait in the protein families that we see. There are only 2 other nucleoside kinases known to exhibit such broad range specificity. So those are the 2 interesting characteristics of TANK. And so now I will go into the methodology of how we purify and produce TANK. First of all, we use standard methods to grow up our cells in standard LB-amp. And when they reach an optical density of 0.6 to 0.8 in the leader cultures, we will induce production of the protein with arabinose because our protein is under an arabinose-inducible promoter. Afterwards, we pellet down the cells, and once we have the pellets, we lyse the cells with benzonase nuclease and with lysozyme. And so what this accomplishes is that the proteins and the materials that we're interested in are inside the cells. And so by lysing the cells, we can gain access to that protein. So in order to purify our protein out of that lysate, which at this point it contains not only our protein of interest, but also all kinds of other molecules and other proteins that we are not necessarily interested in. So we will run through 2 rounds of purification. The first round is called immobilized metal affinity chromatography, and it looks a lot more complex than it actually is. We use a special type of resin which will immobilize, hence the name, a metal like cobalt, and the cobalt will then chelate to the histidine tags that are on our protein of interest. And so by doing so, you are trapping the protein in the column while everything else is flowing through. And then to obtain your protein afterwards, you run another compound through the column that displaces the protein that's already in the column, and thereby you obtain protein with a pretty high purity. And we confirm that it's our protein because it's at a molecular weight of 34.94 kilodaltons. Now, this is our first round of purification, and in order to crystallize, which is our ultimate goal, we need to run through 2 rounds of purification because high purity is a requirement for crystallization. So the second round of purification we go through is size exclusion chromatography. You have these columns that, actually, it's missing a half. Use your imagination here and imagine there's a purple half on the other side. So these purple beads are scattered all throughout the column, and what they do is they isolate your protein of interest by size exclusion. And how that happens is that larger molecules will actually bypass the beads, and so they just fall right through. However, smaller molecules will actually get trapped inside the bead as they go. And so, in other words, you see how the green molecules are slowed by the column while the red ones are going at a much faster rate. You see that in the chromatogram here. And because we ran through a prior round of purification, you see one peak, the blue indicating the protein where it elutes between fractions 30 to 45. So that proves that we have a very high purity in our protein. So that's a plus. It's a green light to go ahead with crystallization, which is the fun part, but also the most time consuming. What you do is we use a hang drop method, whereby we hang a cover slip with a droplet of protein. And essentially how this works, I'm simplifying the details, but there is a reservoir solution which acts as a drying agent. And so that will suck out the water from your droplet, because that droplet consists of not only protein but buffer, which has water in it, and also precipitant chemicals. And by taking the water out of that droplet, essentially you are driving up the concentration of your protein. And so once you hit a critical threshold, you will get, hopefully, crystals. And this is a nanoliter droplet, and you can see that those little tiny specks that seem somewhat amorphous are actually crystals. And you could check on that by altering the lens while you're looking and checking for diffraction. So once you obtain crystals, you want to move on to X-ray crystallography. However, you guys are all scientists. You know that good science requires repetition. And so currently, although I have successfully expressed and purified protein and succeeded in crystallizing in nanoliter quantities, we want to optimize these crystallization conditions and produce crystals in microliter quantities. So we want to scale up. And by doing so, we could attain more crystals for screening and more crystals for X-ray crystallography. And thereby we could look at which so what crystallography does is you're taking a snapshot of the protein as it's interacting with its substrate. So we want to capture our protein as it's interacting with its nucleoside substrates, and then identify which amino acid residues are directly involved in not only the nucleoside activity, nucleoside phosphorylating activity, but also the broad specificity, because those are the 2 anomalous characteristics that we're interested in. So that was where I was at and where I'm heading in the future. And I would like to acknowledge my PI, Dr. Linda Columbus, for her wonderful support, as well as the rest of the Columbus and also the Mirror Labs. And also the generous donors that make the CSS stipend possible. and the UVA Parents Funding Committee, as well as the directors and the participants of the College Science Scholars Program. Y'all are great.
All right, we have time for questions. Anybody? Yeah, here.
What is the role of TANG where?
So nucleoside kinases, that's a good question. They phosphorylate nucleosides, and so they're involved in pyrimidine and purine biosynthesis within the cell. So like nucleosides turn into nucleotides, right? So they help catalyze that. But actually, that's a good point, because broad specificity nucleosides are really not understood, because why would a protein benefit evolutionarily from having such broad range specificity like that? Proteins are supposed to be specific, that's like their job. So that's an interesting thing that people are working on now.
And I'm not sure if you mentioned this or not, but is TANG a human-specific protein?
It's from the Thermoplasma acidophilum, so it's an external file. Yeah.
Do you think that has something to do with its promiscuity, or the fact that these sorts of proteins aren't found in many other phyla?
The only two other organisms where broad specificity occurs that we know of so far is from the Burkholderia thailandensis and the Methanopaldicoccus genesi. And so those two are also clearly from their exotic sounding names. They're external files too. And so it could be, I'm not sure what the analog would be in humans, but we know that within external files, this trait tends to occur. And even then, like, not very frequently. So it's just an interesting anomaly that people are trying to look into, because it provides a glimpse into the whole overarching goal, again, is to better understand proteins and their structure-function relationships. So we want to look at amino acids that pertain to these relationships and kind of tease out those details, if that makes sense.
Great, thank you, Jessica.
While George gets set up, I want to invite you all to get some more food and drink. This would be a good time to stretch too, so please feel free to do that.
I was
I would really like to. Yeah. And don't press it too. I mean, not like press it.
Absolutely. You better hurry.
oh God. You hate that, right? Thought about it.
Okay. Our next speaker is George Schwartz, who is a third-year college science scholar. His mentor at William and Mary in physics is Professor Rosa Lukashu in material science and surface characterization.
Thanks, Karen, for the introduction. So as you said, I'm a third-year college science scholar. My name is George Schwartz. And this summer, I worked at the College of William & Mary for the investigation of surface plasma resonance of silver films capped with cesium oxide layers. My advisor was Professor Rosa Lukashev, and I worked with fellow graduate student Xiaozhu Li and Matt Heimberger. So first off, I want to go in a bit about what surface plasma resonance is. For those of you who don't know, surface plasma resonance is the oscillation of electrons in a metal that results in this evanescence wave that only occurs between the boundary of the metal and dielectric surface. As you can see on this diagram on the far left, this would be a sort of example of what this evanescent wave would look like between these two surfaces. Now, this surface plasma resonance is very determined on very specific conditions. First off, you have to satisfy this dispersion relationship shown through this first equation here. So what this relationship basically shows is that based off the dielectric constants of both the metal and the dielectric, that the surface plasma wave vector can be characterized by following basically inserting into this equation. We did not really consider the permittivity constants, mu1 and mu2 for experiment, so we just assumed these quantities went to 1. Now the other conditions for surface plasma resonance is that the metal portion of the real electric dielectric constant has to be less than 0. In addition, the dielectric real portion of the dielectric also has to be less than the negative of the metal dielectric. These are just properties of surface plasma resonance that have been sort of characterized and known. And from there, then we can move on to start exploring these unique phenomena. So once you have all these conditions met, then you can have an incident wave vector, which is denoted by like K naught, and that can match with the surface plasma wave vector KSP. And note that this surface plasma wave vector is naturally just larger than the incident wave vector. So you need some form of coupler, say like a prism or grating, whenever trying to excite the surface plasma. And this will become apparent when creating this apparatus like that we did this summer. So why is this so interesting? So surface plasma resonance and why we want to explore it was mainly because of that it's very dependent on the properties of both the metal and the dielectric, in addition to the incidence wavelength and angle. So by exploiting this, it then becomes possible that you can measure specifically very specific frequencies in any kind of medium. And specifically for us, we wanted to use it for a metallic photocathode. Now, why metallic photocathodes is that they're very applicable for high energy electron colliders. Yet, the problem is that they have very low quantum efficiencies, something less than 1%, somewhere almost sometimes as low as 10 to the -5th. But they're robust and they have a very small response time. Compared to the semiconductor photocathodes, the reason that those are used so highly is because of their much higher quantum efficiency. So if we can improve this quantum efficiency through surface plasma resonance, then it becomes possible to implement these and has very wide applications in higher energy physics in the future. So now that we brought about you guys to the idea of what surface plasma resonance is, so then we started simulating it and seeing how surface plasma resonance would react given different thicknesses and different incident light. So this is just a basic simulation of silver thickness of 50 nanometer film with increasing cesium oxide thicknesses. And as you can see, based off, as the increasing cesium oxide occurred, the surface plasma resonance would decrease. This is directly relevant because the reflectivity was increasing so that there's less absorption. Essentially, you want a maximum amount of absorption for surface plasma resonance to occur. So in this scenario, we would see that at 1 nanometer with 50 nanometer silver, 50 nanometer silver film with 1 nanometer cesium oxide, you would have the optimal surface plasma resonance excitation. So now that we have the simulation that works, we created this apparatus for this summer with a 405 nanometer laser, modulated 500 hertz that directly calibrated to the lock-in amplifier. And then, as I was saying before, you need some form of coupler in order to excite the surface plasma resonance. And we created this double theta stage that would allow us to make very small changes in the angle, which is very important because, as I was saying before, the incident angle is very dependent. But it changes the surface plasma resonance vector very significantly. So very small changes are important for measurement. So once we have this setup created, then we can start taking results. And as you can see, based off our experiment results, and this is the simulated results from the simulation shown before, that our apparatus actually matches very well with what the simulation tells us. And this is actually very promising because this shows that for silver 40 40 nanometers for 405 nanometer blue light, we were able to accurately not only reproduce the surface plasma resonance, but also make it for multiple trials, but also match it with the specific angle that we would like. And this is a very good initial step from future measurements that we wanted to do with the silver cesium oxide that I was describing before. So this goes into where its consciousness is going in the future. So specifically, we have, first off, you might have noticed that the wavelengths were different from what the simulation I did before and what we actually put in our apparatus. And this is because the cesium oxide, we figure people don't really look into dielectric constant that much for different wavelengths. So that would actually have to be collected by us. In addition to that, we'd have to look into how this alters the work function, because this is very important for the photocathode that I was describing. And lastly, we wanted to look into oblique incidence, because surface plasma resonance is very dependent on how sharp the peak is and where it's located. And by looking at specifically at this oblique incidence and depositing at a different angle, we can improve this sharpness of this peak. So that concludes my talk, and I'd like to thank Professor Lukashev for being an amazing mentor and providing valuable life lessons throughout the summer, my coworkers Xiaozhu Li and Matt Heimerger, and the VMAC Foundation for providing funding for this summer's research, and as always, the College Science Scholars Program. Thank you.
Okay, we have time for questions. Yes.
What does the dielectric constant represent?
The dielectric constant represents a property of the metal or the dielectric. So essentially, how it reacts with an electric field. And if you go in basic EM, it's sort of described as it actually has two parts, a real and imaginary portion, but how the electric wave is going to basically propagate through that medium.
Any others? George, can you tell us a little more about the applications for the metallic photocathode?
Absolutely.
Yeah.
So metallic photocathode specifically, one of the main benefits is that it lasts much longer. It's much more robust and had the response time of something less than a few picoseconds. And also is much more resistant to degradation, contamination, which also is directly correlated to its longer lifetime, which is comparatively to the semiconductor photocathode, which only lasts, say, a few days or months, obviously significantly more impressive. However, because of its lower quantum efficiency, it's not very applied widely, so that's why, this is why we are looking at improving these in the future.
If, as far as using these as sensors are concerned, you'd have to have the light coming in at a very specific angle to
Absolutely,
work?
yes. That's why, that was another unique characteristic of these. Any other questions?
All right. Thank you very much.
Our next speaker is Debbie Pan, who is a third-year college science scholar. Her mentor is Dr. Anindya Dutta in the School of Medicine, Biochemistry, and Molecular Genetics in the topic area of molecular genetics.
Thank you, Karen. Hi, everyone. My name is Debbie Pan, and I'm a third-year science scholar. This past year, I've been working in Dr. Anindya Dutta's lab, as she mentioned, in the UVA School of Medicine, in the Department of Biochemistry and Molecular Genetics. My project has mainly been dealing with a new type of DNA, which I will go into very shortly, but this project is titled Characterization of MicrodNA Stability Using an Artificial Mini-Circle Construct System. All right, to begin, I just want an overview slide for you to follow along with my presentation. This project primarily is a methods-based project, so we'll focus heavily on methods and findings. Of course, I will discuss any sort of results that I've gotten this past year. To begin, what is microDNA? So in the summer of 2012, the Dutta Lab discovered a new type of extrachromosomal circular DNA, which we termed microDNA. So the unique part about this new type of DNA is that it's not a new type of extrachromosomal DNA. We know there's mitochondrial DNA, which is outside the nucleus, but this is a special type of extrachromosomal circular DNA. There have been other ones also, which I won't go into, but primarily the characteristic of an extrachromosomal circular DNA is that there are circles formed outside of chromosomes, and they're typically 2,000 to 20,000 base pairs long. So you can tell why this discovery of circular DNA that are 200 to 400 base pairs long was so exciting. Also, a unique characteristic is that they're derived from non-repetitive genomic sequences. So typically the extrachromosomal circular DNAs that we know of are usually derived from sequences that are repetitive, such as telomeric regions, which are regions of the chromosome that have repeats that minimize the loss of genetic material per replication cycle. So as of last year, the origin and function are currently unknown for this type of DNA. And you can see here, this is pulled from the paper, embellished with a couple of fancy fonts, but these are actual pictures of microDNA that were found in human cells. So one of the ways to tackle what is the function, what is the origin of microDNA is to examine the molecular characteristics of this type of DNA. So my mentor suggested that we create artificially the sequences of a microDNA and see if we put them into cells, whether or not they can persist, whether or not they can replicate or just stay in the cell in order to examine the stability of this construct sequence. I followed that up with a half-life determination experiment of these artificial microDNA. And half-life is just a term that represents how much of a material you have after a certain period of time. So this period of time would be when you have 50% of the material persisting. I was able to introduce this microDNA into two different types of cell lines, U2OS and HeLa cells. U2OS is an osteosarcoma-derived cancer cell line, whereas HeLa cells are derived from cervical cancer cell lines. So we have a bit of interpretation whether or not cancer is involved in this course. So to begin with experimental methods, I first selected a particular sequence of microDNA that we had discovered previously, but this sequence I derived from a DT40 genome, which is actually a chicken cell line, so that there wouldn't be any confusion once we introduce it into human cell lines where the sequences came from. The one I selected was from chromosome 12, and it represented 180 base pairs long at this locus. The second step is to use a method that had been published previously in this journal in 2008 called ligated assisted mini-circle accumulation, which I will refer to as LAMMA from here on out. LAMMA really relies on the torsional strain that can be induced from complementary base pairing. So if you can focus here on the red strand and the green strand, these are just double-stranded DNA sequences, but they relate to each other in the way that the red and the green is simply the red strand sequence split in half, but with their halves reversed and reverse complemented. So if you can see here, if you add the red strand and the green strand together, the red will bind to the second half of the green and form a circle where there are two nicks, one in the red section and one in the green section, so they will force each other into a double-stranded circle. I'll refer to this image later as well. So after isolating that sequence from the DT40 genome, I conducted a restriction enzyme digest in order to isolate that sequence from the original plasmid. And verifying it on an agarose gel, we can see that I did successfully take out this sequence from the plasmid, having the plasmid way over 1,000 base pairs long stay higher up on the gel because it is bigger inherently, so it will travel slower, and the 100 base pair right under the 200 base pair mark. So after the sequence was generated, we have to clone the fragments that would make up the green sequence. So this cloning takes place in an empty PUC19 vector, which was transformed into bacteria and then linearized. This cloning method is called in-fusion cloning, and it relies on having a linearized vector from which you can add fragments. In this case, we'll have two fragments, one from the first half of the green, the other one from the second half of the green, and it will ligate all of them together in a circle. So ligation is simply just the addition of different pieces together based on the homologies between these regions. These fragments were generated by PCR, and as you can see here on another agarose gel, each of these fragments were approximately 90 base pairs long, which means that these fragments were intact and ready to be pieced together. The last step is transformation of all of these fragments into E. coli. The transformation efficiency I got was 80%, which means that 8 out of 10 of these bacterial colonies contain the sequence I wanted. If you can see here, sequencing confirms that the first half of this red sequence was able to bind with this reversed second half of the green sequence. You can see here complementarily TTAACC binds to AATTGG, forming that double-stranded circle. Finally, the circles were confirmed to exist as double-stranded, single-stranded, and this is the linear fragment I started off with first. Inherently, double-stranded are bulkier, so they'll stay higher up on the gel, and single-stranded will go lower. So finally, transfection into U2LS cells requires that these microDNA were transported into these cells using a liposome method, which just encapsulates the DNA and puts it into the cell. After 72 hours, I did 24-hour harvests where I collected all the DNA and then analyzed the quantity with a method called qPCR. Finally, the results show that after 24 hours, 48 hours, and 72 hours, you can trace the half-life back by plotting it on a graph and using this equation to turn it into the half-life calculation. So my conclusion is that microDNA in U2OS cells have a half-life of 9.55 hours. Currently, this semester I've been working on transfecting and carrying out the same exact procedure I described in HeLa cells. And for future considerations, we don't know if this necessarily represents what happens in a human body because this is a test tube experiment. Finally, I would like to thank Dr. Ninya Dutta, my mentor, for the past year, as well as the College Science Scholars Program, and the directors and all of you here who are listening to my presentation. Thank you.
Okay, we have time for one question if anybody has one. Yeah.
MicroDNA, are they found in naturally in all human cells, including like cancer cells?
Right, so what the lab has discovered that they're in most human cells, especially lung tissue and I believe kidney tissue. Right now, we haven't been able to characterize them over all types of human cells.
Great, thank you, Debbie.
So our next two speakers, unfortunately, are both ill, and so they were unable to participate today. Yeah, it's very sad. We are talking about doing a similar kind of event in the spring, and hopefully the two of them would be able to participate at that time. So our next speaker is Leah Grandy, who is a fourth-year CSS student and a DMP in psychology, I will say. Her mentor is Professor Joe Allen, who's in the back. We're glad you're here. And the topic area is clinical psychology.
Hi, my name is Leah Grandy, and today I'll be talking about my psychology research on predictors of change and attachment from adolescence to adulthood. So we know that humans are biologically hardwired to become attached, but what my research is looking at is the quality of these models of attachment. So some people expect that their caregivers will be there for them when they need them. And when asked about their relationship with their parents, they can give a very balanced and coherent description. And these people we would describe as being securely attached. However, other individuals can come to expect that their caregivers will not provide consistent support, and this can lead to maladaptive patterns of thinking about attachment relationships. And these people we would describe as being insecurely attached. So models of attachment are initially based off of your relationship with your parents. But as you get older, they come to include very close peer and romantic relationships. So researchers have identified many of the positive outcomes of having a secure attachment style. For example, lower reports of internalizing disorders such as depression and anxiety, lower levels of externalizing behaviors such as delinquency, risky sexual behavior, and substance abuse. And we also know that the securely attached individual is able to form higher quality, lower stress, peer and romantic relationships. So we really know a lot about how important having a secure attachment style is to your present behavior and also to future outcomes. But what we don't know a lot about is how attachment security can change, which it does. Specifically, we don't know what factors predict attachment discontinuity over a long time period. So, since attachment change research is by nature longitudinal and observational, we cannot directly infer these causal relationships. However, being able to identify these potential causal factors has really important therapeutic implications. So my goal is that researchers can identify a pathway to future security for the insecurely attached individual. So in my study, I used a longitudinal community sample of 130 adolescents who were recruited from a public middle school. And we measured attachment via the adult attachment interview at around age 14, and then again at around age 24. So the adult attachment interview is a 1 to 2 hour interview consisting of questions about your relationship with your parents, and it's observationally coded for how coherent you are thinking and talking about attachment relationships. So I was looking for factors that predicted attachment security at age 24 over and above what was predicted by attachment security at age 14. And I also counted for gender and income. And so the variables I was looking at were all taken from ages 13 to 15, which was roughly when the first AAI was collected. So here are my results. Overall, we found that our adolescents experienced a decrease in attachment security. So the mean value for attachment security at age 24 was significantly lower than the mean value for attachment security at age 14. And this might simply be reflecting that going through adolescence is really tough. And because you have more than just parental relationships, but also peer and romantic relationships feeding into the attachment system, there's a lot of room for things to go wrong. So my findings can be grouped into 4 main categories. So we have positive and negative parental factors and positive and negative peer factors. But I'm not going to go through all of these today. Instead, I'm just going to talk about one negative parental factor and one positive peer factor, just to give you a taste. Okay, so one of our findings was that early interparental hostility predicted relative decreases in attachment security over time. And as you can see, this is accounting for attachment security at age 14 and also gender and income. So overall, this model predicts about 38% of the variance in attachment security at age 24. So how did we get this measure of interparental hostility? So this was the mother and father's reports of their own and their partner's psychological aggression directed towards one another. And so one possible way to explain this is that interparental hostility models maladaptive ways of relating in close relationships, which the teen may then go on to imitate with their peers. And this would explain why interparental hostility is predicting change in attachment security over time, instead of only just being correlated to attachment security at age 24. One of our other findings was that early friendship quality predicted relative increases in attachment security over time, again accounting for these covariates. And overall, our model accounted for about 28% of the variance in attachment security at age 24. And so this measure of friendship quality was taken from the teen's best friend and also a second peer in their friend group, their reports of their friendship quality with the teen. Specifically, these measures were looking at things like emotional support, validation, and caring, and these are all attachment-like qualities. So if a teen is able to form these attachment-like functions in their close peer relationships, this could give them the space they need to kind of step back from parental relationships and reevaluate some previously held concepts they have about attachment. So these results should be interpreted with caution as this study was exploratory in nature. If these findings are replicated, I think they have really important implications for how clinicians should go about treating teens and their friends and families. For example, by really targeting and treating interparental hostility, and also helping teens to form close peer relationships. In the future, I hope researchers will look at attachment change in at-risk samples, and also look at the influence of romantic relationships during this period. I'd like to thank Professor Allen and also Joey Tan and everyone at the Cliff Leader Project for all of their help and support. I'd also like to thank College Science Scholars and all of their generous donors for allowing me to present today.
All right, we have time for questions. Yeah.
I'm pretty sure that I read somewhere that your attachment style as an infant goes on. It also goes on to create like a certain parenting style, and like securely attached infants will become like autonomous parents or whatever the relationship is. I think something like that. Yeah. And so, did you take into account that, like the parenting style, when you were assessing attachment of the teenagers?
We didn't measure these teens' attachment when they were infants, but they are related, but they're not always as related as people think. So the adult attachment interview is really measuring whether you as a parent can provide consistent support, like you as a parent can cultivate a secure attachment style for your infant. So it's kind of two different ways of looking at attachment. But they are related, but we're finding out that they're not quite as related as people have thought.
Was the overall level of attachment and security higher or lower for males or females generally 10 years out, or was it about the same?
To be honest, I'm not really sure. Do you know?
It's generally about the same. Females increase slightly more over time than the males do over this period, but there's not a big difference.
Well, I have a question if you have time. Yeah. So you assessed at age 14 and age 24, and these individuals are going through a lot during that time period, right? They're going through high school, they're going through college, and then they maybe have some post-college years. Mm-hmm. You said there was a decline, slight decline. is this linear or is attachment bouncing around? Like does high school have a bigger effect than college, or or the other way around? Do you know anything about that?
Right.
Yeah.
No, I don't.
Okay.
To be honest, I don't. One of the things I really want to work on after this is to look at how these factors are mediating stuff throughout the time period. So is it kind of like a snowball effect, like gradually these factors become important? So kind of what's going on in the in-between, but right now, that's a really good question.
Thanks. All right, thank you.
Okay, our last speaker for today is Rebecca Walker, who is a fourth-year college science scholar. Her mentors are Professor Howie Epstein and Professor Erin Mills from our Department of Environmental Sciences, and her topic area is ecosystem ecology.
Hi everyone, so as Professor said, my name is Rebecca Walker. I'm a fourth year, and I will be presenting my research on nitrogen cycling through forest succession. I feel like we're really moving up in scale from the micro. Now we're looking at the ecosystem scale. So this project arose out of a larger study looking at carbon dynamics in the Mid-Atlantic. So currently the Mid-Atlantic is experiencing a period of forest regrowth following agricultural abandonment. So in this model on the left, the green represents primary forest, the red represents agricultural land, and the blue represents secondary growth forests. So as you can see at the start of the model, the majority of the Mid-Atlantic and the East Coast is primary forest. So through the centuries, we see agriculture develop, and then eventually secondary forest follows. So today, the majority of the Mid-Atlantic is dominated by secondary growth forests. These stands range in age from about 50 to 100 years, and globally, and as these forests regrow, they're removing carbon from the atmosphere and incorporating it into their biomass, and this is referred to as carbon sequestration. So globally, regrowing forests accounts for approximately 28% of carbon sequestration. And these regrowing forests play a key role in the United States' carbon budget by offsetting a portion of our emissions. However, there remains much uncertainty about how long these forests will continue to sequester carbon. So the goal of our study is to address this uncertainty by looking at carbon dynamics in old-growth forests of the Mid-Atlantic. So carbon is incorporated into biomass through photosynthesis. And because nitrogen is a key element in photosynthesis and the limiting nutrient in the Mid-Atlantic, I chose to look at nitrogen dynamics through forest succession. The prevailing model of nitrogen through forest succession is that the total nitrogen pool will increase as the stand ages and more nitrogen is incorporated into stable forms of organic nitrogen, so soil organic nitrogen. On this graph, the x-axis shows the age of the forest stand, and the y-axis shows the nitrogen pool. And as you can see, nitrogen is increasing with stand age. However, this data stops at about 180 years, which is the very beginning of the old growth stage. So we don't really know what happens in this later stage of forest succession. So based on this, I wanted to address 3 questions. All of our questions are concerning nitrogen and the organic horizon. And the organic horizon is the top soil layer made up of decomposing biomass from plants and animals. So the first question is, does the total nitrogen pool and the organic horizon increase with stand age in the Mid-Atlantic forests? Next, we wanted to determine, do dynamics of organic and inorganic nitrogen change as a forest transitions from secondary growth to old growth? And finally, does a correlation exist between soil nitrogen characteristics and the amount of carbon that's actually stored within these forests? To address these questions, 25 old growth sites were identified in the Mid-Atlantic. At this point, old growth accounts for about 1% of forests in the Mid-Atlantic, so it's a pretty small amount and difficult to get to. At each site, we assessed old growth using 5 characteristics. The first is high densities of large old trees. The second is a large volume of dead wood. The third is the presence of canopy gaps, which occur when large trees fall down, or as they age, they fall. The fourth is a multi-layer canopy that occurs as younger trees begin to grow up in the canopy gaps. And the fifth is the lack of evidence of human disturbance. So no things like fences or cut stumps, things of this nature. And at each of the sites, we sampled above ground dead and live biomass, as well as collecting leaf litter and soil samples from the organic horizon. And soil samples were collected from the old growth sites and paired secondary growth sites with similar site characteristics. Further, we collected tree cores. That's my graduate student in Virginia and Garvey. We collected tree cores in order to assess the age of the old growth sites. And then the soil samples were subsequently analyzed in the lab for our 4 nitrogen characteristics, which were nitrate concentration, ammonium concentration, which I'll refer to as the inorganic variables, and then percent nitrogen and the carbon to nitrogen ratio, which is the ratio of these 2 elements in the soil, which I'll refer to as C to N ratio. So now to return to our original questions. Does the nitrogen pool in the organic horizon increase with stand age in mid-Atlantic forests? In short, no. So this graph shows stand age on the x-axis and the organic horizon nitrogen on the y-axis. And as you can see, we saw a significant decrease in the nitrogen pool with stand age in the old growth stage. So this is just our old growth data. And this is counter to the prevailing model, which predicted an increase in nitrogen with stand age. So from this, it sort of suggests that the nitrogen pool reaches a maximum at about 150 years and then begins to decline through the old growth stage. However, this needs to be taken with a grain of salt because, as you can see, there's a limited number of points at the upper age of our stand ages. Next, how do the dynamics of organic and inorganic nitrogen change as the forest transitions from secondary growth to old growth? It's really looking at these variables through succession. So these graphs each have secondary growth variables on the x-axis and old growth variables on the y-axis. The top graph has percent nitrogen, the middle graph has C to N ratio, and the bottom graph has ammonium concentration. For all 3 of these variables, the secondary growth values were shown to significantly predict the old growth values. So what this means is there's almost no difference between the values in the secondary growth sites and the values in the old growth sites. So this suggests that successional stage is not driving nitrogen characteristics. And instead, nitrogen characteristics are controlled by something varying at a regional level. So this includes abiotic factors such as temperature and moisture of the soil, and biotic factors such as the species composition of the overlying canopy. Next question. Does a correlation exist between nitrogen characteristics and the amount of carbon stored within old growth forest stands of the Mid-Atlantic? In short, the answer to this is no as well. However, we did find one significant correlation, and that was between percent nitrogen and the soil organic carbon pool. So that's basically the amount of carbon stored in the soil. So on the x-axis we have percent nitrogen, on the y-axis we have soil organic carbon, and you can see that carbon is increasing as nitrogen increases. So because this is a percent nitrogen increase, this suggests that the proportion of microbes in the soil is increasing as increases as soil organic carbon increases, which suggests that carbon storage in the soils is mediated by microbes. And this is really important because often within a forest, the largest store of carbon is in the soils. So in conclusion, the data suggests that decreases are basically, because our soil carbon pool is significantly related to this percent nitrogen, which, as we said, would indicate a microbial mediation, and because percent nitrogen was found to decrease with stand age, this suggests that the ability of soils to sequester carbon in the old growth stage decreases. Second, we also found that site-specific characteristics that vary on a regional level are really driving our nitrogen dynamics. rather than successional stage. So, in conclusion, these two factors have important implications for management strategies for both nitrogen and carbon for these forests. I would like to thank my mentors, Professor Howard Epstein and Aaron Mills, and also my graduate student Jennifer McGarvey, as well as the National Science Foundation and the College Science Scholars for their generous funding that made this research possible. Thank you all so much.
All right, we have time for a question. Anybody has one? Yes.
Did you determine which either biotic factors or abiotic factors cause the regional disparities in the nitrogen?
So we looked at some of this, and again, in short, no. We ran a test that sort of looks at a lot of these variables at once and looks to see if some of them are driving another one, and we didn't find anything that was particularly significant. So a lot of these things need different data needs to be collected in order to really tease out what's causing it. And also, it's difficult to pinpoint one factor because they interact so much together. So the temperature and wetness can influence microbial activity. So it's difficult to parse out that specific thing when really it's a combination of things.
Anything else? Yeah?
Different species of trees have any effect, or just one type of tree?
We did look at species composition. I didn't include that graph, and it does influence some of the variability we see in percent nitrogen, the ammonium concentration, and the C to N ratio. So yes, the species have an effect, definitely. So that's one of the regional factors we would consider for sure.
All right, thank you.
Okay, I want to thank all of our special guests and all of our special student guests for coming today and supporting our students. I want to give one more round of applause to our amazing science scholar presenters today. I'd like to invite you to keep munching along with us and visit for a little bit. We have plenty of time, so you can ask any of the questions that were burning inside of you and there wasn't time for. Now you have a chance.
Are you guys sitting down?